Hi class, let's get started with our discussion on viruses, those enigmatic subatomic or submolecular particles that are smaller than a cell. So, how do we look at our viruses? Where do we put them on our biological or living spectrum? And they're kind of tricky to categorize. They're so small that there's a lot of debate about whether they are or are not alive. We'll get to that later. I don't want to get ahead of myself. So first, let's talk about what viruses do. They infect cells. All viruses will infect cells. Without a cell to be infected, the virus will cease to exist. They can infect any kind of cell, from a bacterial, an archaeal, and fungal, prozoal, plant, animal, so on and so forth. Seawater has the highest concentration of viruses of any water source. They have an incredibly high unit count of viruses. Thankfully, these 100 million viruses per milliliter of seawater are mostly going to fall into the bacteriophage category, or viruses that are designed to specifically attack the bacteria that they come in contact with. For a very long time, the cause of our viral infections wasn't very well known. The reason for this was we could not see them with a light microscope. And if we can't see them with a light microscope, it's going to be really, really tough to identify them and properly isolate them. Louis Pasteur postulated a long time ago that there's some living thing smaller than the bacteria that was in the liquid or the supernatant that's filtered off from infected hosts. He proposed the term virus, which is the Latin term for poison. Later on, Ivanovsky and Beranchik showed that the disease in the tobacco plant was caused by a virus. The specific disease was tobacco mosaic, or the specific virus was tobacco mosaic virus. Lothar and Frosch discovered that animal viruses can cause hoof and mouth diseases in cattle, and there are filterable and unfilterable viruses. Our filterable viruses are going to be viruses that can be found when fluids from our host organisms are passed through a filter, and this isn't just a cotton filter or a typical cellulose filter, it's a porcelain filter. And this porcelain filter is designed to trap bacteria. So it's capable of trapping organisms that are on less than 10 microns in size, but greater than a micron. And remember, a micron is 1/1,000,000th of a meter. Viruses, though, are very, very tiny. They're typically going to be measured on the nanometer scale, or the 1/1,000,000,000th of a meter scale. So it's very common for a virus particle, or viron, to be less than 1 micron, and hence it can pass easily through the porcelain filter that will trap bacteria, but the filtrate that goes through that filter will still have the virus particles in it and remain infectious. This filterable virus conclusively showed that something without any cells was capable of containing an infectious agent. And there's still a lot of questions that remain about viruses. They are notoriously hard to culture and study and maintain in a laboratory environment. If they're organisms, if we consider a virus an organism, are they alive? This is kind of a basic question. I mean, if you look at a frog, if you look at a human, if you look at a bear, you can pretty clearly tell, is it alive, is it not alive? But is a virus actually alive? In most biology classes or introductory to biology classes, we have the characteristics of life. They need to be able to have organization, they need to have metabolism, they need to have reproduction, and they need to have responsiveness to their environment. There's all these characteristics that are given for something to be alive. Viruses don't actually meet all the characteristics. Another question that we have about viruses is, what role do they play in the evolution of life? Viruses are often associated with modifying the host genomes. As they modified host genomes, did they cause new species to develop? Did they cause new breeds to develop? How has the change of our host genomes by viruses impacted speciation throughout time? What are distinctive biological characteristics of viruses? And how can a particle that's so small cause such a big problem? This is more of a rhetorical question. This has been fairly well answered at that point. And then finally, what connections are there between viruses and cancer? We found a few, but are there more? Are there additional connections that we have not yet identified that we can exploit in the ever-present, ever-popular war on cancer? So let's talk about this viral debate. Since viruses are unable to replicate independently from their host cells, many people do not think they should be called living, because one of the characteristics of life is being able to reproduce. and viruses cannot reproduce by themselves. And instead of calling them organisms, we should call a virus an infectious particle. And depending on the literature that you read, many times you'll hear a virus referred to as, instead of a virus organism, you'll hear it referred to as a viron particle. Ooh, good coffee. Anyway, where was I? All right, so viruses do exhibit most of the processes of cells, and they are capable of reprogramming cells. So viruses are definitely going to be more than just an inert molecule, an inert life form or lifeless piece of atomic mass. So what's the difference? Is there some in between or spectrum between living and not living? So I may slip up and say that viruses are alive and not alive, particularly when we get to our sterilization techniques. But the best term, the technically correct term, is to say that they're active or inactive. And that's a middle ground that everyone can agree on. This virus is active, it's capable of infecting the cell and reproducing, or this virus is inactive. This virus is incapable of infecting the cell and reproducing. As viruses will infect cells, they are going to change the genome of cells. This has a very significant role on the changes on the genomes of organisms. It's been hypothesized, or estimated, I should say, it's been estimated that approximately 10% of our human genome is made of sequences that have come from virus particles. And if we look at bacterial DNA, it's been found that 10 to 20% of bacterial DNA contains viral sequences depending on the species of bacteria. So because these viruses depend on our hosts, on the host cell for reproduction and incorporate their DNA into the host cell's machinery, we can think of a virus as an obligate intracellular parasite. That's kind of a $10 term. Let's talk about these individual words. Obligate means that it is necessary, it is mandatory. The virus is obligated to this form of reproduction. Intra is a prefix for intracellular. Intra meaning inside. Inside of what? Inside of the cell. And then parasite meaning that this virus particle benefits, but the host cell is at a disadvantage. So this active virus particle cannot multiply unless it's in the specific host cell and reworking the genetic machinery and the metabolic machinery in the host cell to make and release new viron particles. The virus must have the host cell, and it must get into the host cell. So here's a table from your text summarizing some properties of viruses. If we look at viruses, they are ubiquitous. They are everywhere. They are very, 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 very tiny. They can range from as small as 20 nanometers to up to 450 nanometers, approximately half a micron in size. There's a couple exceptions, but generally speaking, the vast majority of viruses fall into that size range. Our viruses are going to have either DNA or RNA for their genetic storage information. They can have either single or double-stranded DNA or RNA. Our viruses lack enzymes for most of their metabolic processes, but that's okay because they use the cell's enzymes. And viruses also lack the machinery for synthesizing proteins, so they don't have any ribosomes in them, but that's okay. The virus will use the host cell ribosomes. For a long time, when we named a virus, we named the virus based on the host that it was found in and the disease that it's caused. And that's a pretty straightforward naming system. Think of the tobacco mosaic virus. The tobacco mosaic virus is a virus that infected tobacco plants, and it caused little colored spotches or splotches to appear on the skin, on the leaves of the plants. It looked like a mosaic. The newer classification system still takes into account the host and the disease, but also looks at a more structural or chemical or composition viewpoint of the virus, or the genetic makeup of the virus. So now we're able to find virus subtypes and more accurately categorize our viruses based on their storage information. Are these going to be a double-stranded DNA virus? Are these going to be a single-stranded RNA virus? The genetic makeup is a very important way of categorizing the virus and can strongly influence the rate of mutation of the virus and strongly influence how we target the virus. What kind of antiviral medications would be effective? Since the new naming system has been instituted, the International Committee on the Taxonomy of Viruses has categorized viruses into 3 orders. Within those 3 orders, there are 75 families, and within those 73 families, and within those 73 families, there are 283 genera, and then within those genera, we have the individual species of viruses. So, let's take a concept check here. Which of the following best describes a virus? Is it a heterotrophic organism? Is it saprobic? It's an obligate intracellular parasite? Is it chemoautotrophic? Or is it photosynthetic? If you do not remember, pause the video and flip back through your notes or rewind the video. Alright, the answer is C, obligate intracellular parasite. If you have any questions, please shoot me an email or post them on the discussion board. Happy studies!